Hello students, how's you? I hope you all are well. In the last class, we have discussed about the semi-autonomous organelle. We discussed about the mitochondria and a little bit we also discussed about the chloroplast and we discussed the structure of the chloroplast. And we already know plants do not perform photosynthesis in all light. They perform photosynthesis in a visible light, that is the photosynthetic active radiation. If I say that is the ranges are up to 380 to 750, that is the exceed 400 to 700 nanometer as with here blue and red light. Plant perform maximum photosynthesis, green plant in the blue and red light, and they perform minimum photosynthesis in the green light because green reflect back to each other. So this structure that we read in the last class. Yes. So we know chlorophyll is of pigments are of different types. That is the chlorophyll A. We have the chlorophyll A chemical formula, that is the C55H72 magnesium N4 and O5. Whereas the chlorophyll B, we have the C55H70 magnesium N4 and O6. Now there is a one difference, that in chlorophyll A there is a methyl group. What is that? There is a methyl group at the second position. Whereas in the chlorophyll B we have the aldehyde group. What is that? We have the aldehyde group at the second position. So if we talk about the chlorophyll is a derivative of porphyrin. Now porphyrin, that is the central chemical structure of chlorophyll has a magnesium atom. Do you know the chlorophyll structure resembles to the hemoglobin because in the center we have the magnesium atom and 4 nitrogen atoms are found around the magnesium like this. There is a pyrrole ring structure with 4 carbon around the nitrogen. So we have the pyrrole ring around the nitrogen like that. And a hydrocarbon end is also seen. The 4 pyrrole rings with the nitrogen and the magnesium are together termed as the porphyrin head. One we have the porphyrin head and the hydrocarbon end is the phytol tail. So this is the phytol tail. What is that? Phytol tail. This is the porphyrin ring. What is that? Porphyrin ring. Now along with chlorophyll A, B, we have the other accessory pigments, that is your xanthophylls as well as the carotenoids. Now if we want to see these pigments, so how we can separate this pigment with the help of the chromatography using the acetone and paper. So when we will do the chromatography, so we will observe the different pigments. So when chlorophyll A gives the bright and blue-green color in the chromatograph, chlorophyll B gives your yellow color and xanthophyll gives you yellow color, carotenoids gives you yellow to yellow-orange color. This is all about the chloroplast and the pigment that is present in the chloroplast. Now the second question that has been asked in the UPSC, the functions of the ribosomes and the subunits. So firstly we have to understand what are the ribosomes. So ribo is the ribonucleic acid. Now zomes, what is the literal meaning of the zomes? That is the bodies. So these are the complex molecular machine present inside the living cells. What are these? Living molecular machines found inside the living cell. what is the main function if we say? So these ribosomes are the protein factory. Protein factory. They form protein. Now ribosomes are associated in both prokaryotic as well as the eukaryotic. It is the single organelle that is present in the both prokaryotic and eukaryotic. Present in prokaryotic as well as the eukaryotic. Now they could be lie in the cytoplasm, or they could be attached to the membrane, or could be attached to the nucleus or endoplasmic reticulum surface. Now who observed the ribosome the first time? That is the George Pellet. Who's that? George Pellet. George Pellet observed the ribosomes for the first time, and they are the very tiny dense spherical bodies that we have 150 to 200 angstrom, that is the diameter. And I already told you these are the protein sites, and in prokaryote they lies in the cytoplasm in freely. In eukaryotic they lies in the cytoplasm in freely, as well as in the eukaryotic cell they are also present in the mitochondria and the chloroplast. Mitochondria and chloroplast, or they are attached to endoplasmic reticulum. So that's why we have two types of the reticulum, that is the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Rough because ribosomes are attached, and RER is also involved in the protein synthesis. Now let's move to the structures of the ribosomes, that is the structures of ribosomes. Now when we say structure of the ribosomes, it is made up of the complex, that is the RNA, rRNA and proteins. Now where the ribosomes are formed? They are formed in the nucleolus. Nucleolus is present in the nucleus. So rRNA plus proteins. So that's why we call it as the ribonucleoprotein complex. What is that? Ribonucleus protein complex. Yes. And the amount of RNA and protein varies in eukaryotic and prokaryotic cell. This varies in pro and eukaryotic cell, as well as the proteins also quantity varies. So we have the larger subunit and the smaller subunit. Firstly, let's talk about the prokaryotic cell, in primitive prokaryotic cell. We read that in prokaryotic cell we have the 70S, and in eukaryotic we have the 80S. Now what is the meaning of the S? That is the Swedberg unit. What is that? It is the Swedberg unit. The scientist who discovered this unit is the Swedberg unit, or sedimentation coefficient. Sedimentation coefficient means when they took the units and centrifuged, so what they found? Less density means less sedimentation unit. More the density, more will be the sedimentation unit. So when we say 70S, that is in the prokaryote. So 70S is again made up of the small two subunit. One will be the larger subunit. One is the smaller subunit. I told you these are made up of the rRNA and the proteins. So in 70S we have the 50S, that is the larger subunit, and 30S, that is the smaller subunit. Clear? Yes. Now again these 30 and 50S are made up of the again smaller subunits. So when we centrifuged again these units, larger and smaller subunits, so we found that (23S), 16S, as well as we have the 5S rRNA. Now your your 5S rRNA, that is present in the larger subunit. And these are in the smaller subunit. And how many nucleotides are present in these? So when we talk about the 16S RNA, so we have it is made up of the 1650 nucleotide. When we talk about the 5S RNA, so it is made up of your 120 nucleotide. And your last that we have the 23, that is made up of the 3300 nucleotide. What is that? 3300 nucleotide. This is all. Now when we talk about the larger subunit, let's talk about the larger subunit. So larger subunit, not larger subunit, we are talking about the 80S, that is in the eukaryotic. So it is made up of the two units. One is the larger subunit, one is the smaller subunit. The larger subunit, that is the 60S and 40S. Now one thing I forget to tell that you were saying that 60S and 40S is the 100. So why are you writing here the 80S? Remember, don't use the second class maths. These ribosomes units are never additive. They are not additive in nature. Yes. So these are made up of 40S. That is your 40S is made up of the 30 proteins, as well as the how many ribosomes are, that is the 18S RNA. When we talk about the larger subunit, so your larger subunit, that is made up of the 28S rRNA, 5.8S rRNA, as well as the 40. 28 we have, 5.8, that is and 5S RNA. Yes, 5S RNA. sRNA. Yes. So these 28 as well as the 5S and 5.8, they are present in the larger subunit, and 80S, that is present in the smaller subunits. And larger subunit is like a dome shape. It contains the channels where plays very important role during the translation, that is the formation of the proteins. So if we talk about in the eukaryotic cell, so in eukaryotic cell we have the 70S ribosomes. How much? 70S ribosomes. In mitochondria and chloroplast. And how these two subunits are present in a cell? So these two subunits are not present like together. They are not bound together like this. Like one subunit will be like this, dome shaped, and one shape will be flat. So they are lies freely in the cytoplasm. Freely in cytoplasm. During the protein synthesis, they will come together, and how they will come, it depends upon the magnesium ion. Higher the concentration of the magnesium ion, they will join together in a cell. And if we talk about the molecular weight of the 70S and 80S, so the 70S molecular weight, that is the 2.7. That is the 2.7 into 1.6 Dalton. If we talk about in the 80S, so the molecular weight, that is the 40 into 106 Dalton. Yes. So there are some things that you need to remember. These ribosomes units vary in the every cell. So mitochondria of fungi. Mitochondria of fungi. So the ribosome units, that is the 77S. When we talk about the mitochondria of mammals or animal cells, mitochondria of mammals and animal cells, these are the 60S. As we used to study in lower classes that it is 70S is present, but it varies in each and every cell. And in the chloroplast, in ribosomes of chloroplast, that is the 70S. That you need to remember this thing. Yes. So this is all about the units that we discussed about this. Okay. Now let's discuss the little bit functions of these functions. So the first function, that is they assembles the amino acids to form protein. Assemble amino acids to form proteins. Means they are during the translation. This is the larger subunit, and this is the smaller subunit. And you should know about that we're talking about the rRNA. So RNA is of different types, that is the mRNA. We are talking about it is made up of the rRNA and the tRNA. So ribonucleic acids. So ribonucleic acid is of 3 types, and this rRNA is responsible for the formation of the protein synthesis. This is the messenger RNA, from where the amino acids will join together and form the codon or anticodon, and they will form the polypeptide. tRNA, that brings the amino acids from the cell and binds to the site of the protein synthesis. So this mRNA is synthesized in the nucleus and transported to the cytoplasm for the process of the translation. So mRNA, where it is formed? Nucleus. Then transported into the cytoplasm for translation. And during translation, who will help? Ribosomes will help. Larger and smaller subunit will help. So in short, we can say that the ribosomes are the sites of the protein synthesis. So ribosomes will bind around the single mRNA. And you need to remember this thing also, that ribosomes in prokaryotes, during the translation, they bind, lot of ribosomes bind and they form different kind of the proteins. So this chain, this is the mRNA. These are the ribosomes. So lot of ribosomes attached to a single mRNA, and this is known as the polyribosomes. What is that? Polyribosomes. But in eukaryotic, only the one type of the protein synthesis at a one time. So that's all for the day. Thank you and have a nice day.